The complement system. Let us now study pathways of complement activation. The first pathway which we are going to study is the alternative pathway. The alternative pathway. Scientists discovered it second. That is, alternative pathway was discovered after classical pathway, hence the name. Alternative pathway is one of the first responses of the innate immune system to the invading pathogen or infection. Let's study the steps involved in the alternative pathway. We must remember that C3 is the most abundant protein of the complement system. In the first step of the alternative pathway, this C3 undergoes spontaneous hydrolysis in the blood, yielding a molecule which is represented as C3H2O. The hydrolysis of C3 actually opens or reveals a site in the molecule to which the next protein of the alternative pathway binds. In the next step, factor B comes and binds to the C3H2O molecule. Further, factor D comes and cleaves this factor B into larger and smaller fragment. The larger fragment represented by BB remains bound to the C3H2O molecule, whereas the smaller fragment BB is released. Now, as a result, C3H2O-BB complex is formed. This complex has protease activity. It specifically and rapidly cleaves and activates many more molecules of C3. Remember, C3 proteins are present in blood in abundance. So C3 molecules are cleaved into C3b and C3a fragments. Now, proteases that cleave and activate C3 are known as C3 convertases. Thus, this C3H2O-BB complex is a C3 convertase, but it is a soluble C3 convertase. When there is an infection, some of these C3b molecules bind covalently to the nearby microbial surfaces. This is known as complement fixation because C3b becomes firmly fixed to the pathogen. In the next step, factor D comes and cleaves factor B, generating C3b-BB complex, which is now located on the microbial surface. This C3b-BB complex has C3 convertase activity, and it is known as alternative C3 convertase, which works at the surface of the pathogen. This complex is unstable, so another protein known as properdin, represented by capital P, comes and binds this complex and stabilizes it. So, we saw that alternative pathway leads to the generation of C3 convertase, which is represented by C3b-BB complex. And this complex is also known as alternative C3 convertase. We must note that all the three pathways of complement activation lead to the generation of C3 convertase. And the terminal steps of these all pathways are same. We will see them later, but let us first move to the next pathway and see how that pathway leads to the generation of C3 convertase.